So today I would like to talk about the process of how prions are developed in the brain and how they control the brain into becoming degenerative and what prion diseases are in general, along with why we need to come up with a solution for problems such as Kuru and mad cow disease or Creutzfeldt-Jakob disease. So prions are misfolded proteins which are present in the brain, and these lead to the proteins with the normal structures that can fold into chromatin and properly code for different types of genes. These misfolded proteins cause almost every other gene around them in their environment to become misfolded as well. And this is a problem because misfolded proteins can't properly carry out instructions in the brain. And so signals and impulses transmitted by the neurons cannot properly be processed throughout the brain, which is why prion diseases are referred to as degenerative brain disorders. And an example that I sought to study was mad cow disease or Creutzfeldt-Jakob disease, which is developed by eating the meat from an infected cow. And this prion disease is actually really deadly, as you will die within 1 to 2 years of actually getting the infection from the meat. And even though there haven't been a lot of cases, there's been about 230 cases since 1996, and Great Britain has 178 of those. There is reason to be concerned about this disease because I think it shows a lot more about the food industry and how there are not regulations being met. And this experiment and experimenting with mad cow disease, like looking at the experimental test results and really trying to seek out the answer of why humans are able to contract something like this from a cow can help us solve numerous other problems in terms of diseases and the causes of some of our other ailments in the present world, such as the different types of cancers, such as leukemia, and etc. So, I would like to take or draw a model of how exactly the prions are regenerated throughout the brain, and so this would help you better like take a picture and a view at it. So there's PRPC, which is then during a disease there's a conversion of the PRPC into basically toxic intermediates and basically forms of PRPSC. Cap. It's like a medicine pill in which there are two different colors that are used to represent this PRPSC, and these continuously then keep developing, and there's an accumulation of this process, and these this process is like the PRPSC is what leads to the neurodegeneration in the brain, and this is the actual infection. So the PRPSC, we have specific parts of it, that I'll represent here in blue. And then there's an overall accumulation, like I said, of hundreds of these PRPSC molecules. So I would just develop this right here. And so this blue right here, D is. And so then there's a sort of a recruitment process. So now, many of you might be wondering what PRPSC exactly is. And so PRPSC is a degenerative disease in the nervous systems of sheep and goats. And it's like a spongiform encephalopathy, which is what mad cow disease is, or VCJD is. And this is the molecule, or the I should say protein, that accumulates in the brain, and what actually leads to our human brain degenerating. And the normal cellular protein is PRPC, and these cultured cells of PRPSC, when they accumulate in the brain, this is what leads to the degeneration and why the disease is so fatal, because a person will die within a span of like 1 to 3 years. So getting back to this model, this was basically a summary of how PRPSC accumulates in the brain, what it is, what prion diseases are, and why they are so deadly. And ultimately, it's because they're misfolded proteins, and they lead to the normal proteins, the normal molecules, PRPC, to be like a mutant form, and this really leads to the brain being degenerative and not having the same capacities it once did.